Hello everyone, I'm Gazelle and I'd like to talk about my project which is about synthesis of technetium radioagents for in vivo imaging of amyloid beta plaques with SPECT. First, I'd like to give a short introduction about Alzheimer's disease. Alzheimer's disease is a central nervous system disorder which can be a result of protein aggregates of amyloid beta, which is extracellular plaques or tau known as neurofibrillary tangles, or generally neuronal injury as it's shown in the picture. It is called 18 classification. Lately, many therapeutic and diagnostic attempts have been made focused on amyloid beta, like inhibition of amyloid beta productions, inhibition of aggregation and deposition of amyloid betas, or amyloid beta immunotherapy, which has been unsuccessful because of the limitation of crossing the blood-brain barrier. With the advancement in research, nuclear medicine imaging of Alzheimer's disease and kindred dementias has taken center stage. Several PET radiopharmaceuticals with carbon-11 with a half-lifetime of 20 minutes and fluorine-18 with a half-lifetime of 109 minutes have been licensed for clinical use. Different examples are like fluorine-18 fluotemetamol, carbon-11, PIB, fluorine-18 fluorobetaben, and fluorine-18 fluorobetavir. These strategies and tracers have their pros and cons, such as they cross the blood-brain barrier and ease the process of in vivo imaging of amyloid beta burden. However, because of the high cost, it might not be available everywhere, especially in rural areas and poor countries. Also, limited half-life time may cause difficulty in the process of synthesis and injection. As is shown in this slide, the other nuclear medicine imaging technology SPECT has been widely available since the early 1990s. As of 2015 in Canada, SPECT scanners outnumber PET scanners by at least 40 to 1. In a vast populated country, access to either of these highly sensitive modalities could improve patients' management, reduce burden on patients and caregivers, and improve therapy outcomes. So it's believed it's time to consider developing technetium radioagents for in vivo imaging of amyloid beta. In this slide, you can see 20 of more than 50 attempts for synthesis of technetium radiotracers, which weren't successful for in vivo imaging because of high molecular weight, unsuitable lipophilicity, high charge, or instability. Many potential technetium agents have been synthesized that fulfill the in vitro evaluation requirements of an Alzheimer's disease imaging probe, namely efficient radiolabeling and stability, affinity for amyloid beta fibrils and selectivity, and binding affinity for amyloid beta plaques on brain tissue, but nonetheless fail the prerequisite for adequate in vivo brain penetration. One of the latest one which is taken for further research is the thiazole and imidazole effort by Sagno in 2019. This paper had acceptable in vivo results because of the small size, low molecular weight, and moderate lipophilicity of the cyclopentadienyl tricarbonyl technetium core. Also, the phenyl moiety of 2-phenylbenzothiazole has been replaced by cyclopentadienyl tricarbonyl, and 2-aryl-benzothiazole is known as the pharmacophore to confer binding affinity to amyloid beta plaques. The affinity of complexes rhenium 1,2,3 for amyloid beta plaques was evaluated with confocal microscopy in fixed brain section from an Alzheimer's disease patient using a standard staining procedure. The figure here is showing the fluorescence microscope images obtained in brain slices and with the amyloid beta binding histological dye thioflavin S as a positive control. All three complexes bind selectively to the amyloid beta plaques, producing images of analogs quality to those of thioflavin S. The brain uptake of technetium-1 is comparable to that of fluorine-18 fluorobetavir with 7.33% injected dose per gram at 2 minutes and higher than that of fluorine-18 fluorobetavir with 4.77% injected dose per gram at 2 minutes. The U.S. Food and Drug Administration approved both imaging agents in clinical use. The brain uptake of 99M technetium 123 far exceeds all values reported for potential technetium amyloid beta imaging probes in healthy mice, and the high brain penetration noted for technetium 123 was accompanied by fast blood clearance and almost complete washout at 90 minutes, resulting in a good clearance ratio, which indicate lack of retention in brain tissue, an advantageous feature for clear imaging and one of the prerequisites for amyloid beta imaging probes. The process of synthesis was started from ferrocyanide compounds as precursors and followed by the synthesis of rhenium complexes as standards. Radiolabeling was done by ligand exchange reaction under the nitrogen flow. Based on the previous result and talk, the synthesis of new derivatives of rhenium and technetium complexes are considered, which may have acceptable lipophilicity and molecular weight to be uptaken and washed out from the brain. One of them is oxazole compounds and the other one is chalcones because of the good results of the other similar papers as well. So far, the synthesis of the compounds in the table has been done and further evaluations and radiolabeling is in progress. At the end, I would like to thank to the source of funding and thank you for listening. I'd be glad to answer your questions in chat.